Thanks very much for the invitation to present. So I was going to talk today, giving some background over the antibiotic resistance crisis and then some possible solutions for how we can develop new antibiotics using some case studies from research that we're doing up at UQ on trying to do exactly that. So the last decade, there's been a lot of popular press articles talking about the antibiotic resistance crisis, and some of the headlines within those articles have been quite sensationalist. One of my favorite is the mop of death. But they do reflect a reality, as pointed out by the World Health Organization, that the world is heading towards the post-antibiotic era, where simple infections that have been treated for the last 50 or 60 years can once again kill. And there are a lot of implications just beyond what you'd commonly think of infections causing death, in terms of a lot of other mainstream therapies would no longer be effective. So things like joint replacements, things like chemotherapy, which rely on antibiotics to keep infections under control, would no longer be possible. And so beyond the health effects, there can be significant economic effects. So it's predicted that if we don't do anything, there could be a 3-4% drop in the GDP by 2050. And that's equivalent to what caused the level of drop in GDP with the global recession. And it's also predicted without new treatments, the number of deaths caused by drug-resistant drug-resistant bacterial infections would be about 10 million patients per year. So antibiotic resistance is nothing new. Every single antibiotic that's been introduced, generally within a few years of its introduction, clinical resistance has been observed. And that goes from the very first antibiotic of penicillin to the most recent ones of daptomycin and ceftaroline. And that applies in Australia as well. So Australia, depending on which type of bacteria, for example, methicillin-resistant Staph aureus, we see moderate levels of resistance, so we're better off than other countries. But on the other hand, for vancomycin-resistant enterococci, we're actually leading the world in the rate of infecting by those bacteria. So what is driving this development of antibiotic resistance? It really is misuse of this valuable drug that was discovered originally in the 1940s. So, for example, if you look at regional variation in prescriptions, and this is data from England, you can see vast differences in the number of antibiotics that are being prescribed per person. And there's no reason why that should differ between regions. And even more strikingly, if you look at seasonal variations in prescriptions, you can see that in the English summer, very few antibiotics are prescribed, whereas in December, in the middle of winter, of cold and flu system, you get this huge increase in antibiotic prescriptions. And, of course, colds and flus are not able to be treated with antibiotics. So you have this large unnecessary increase in prescriptions. The other major use of antibiotics, and actually 70% of antibiotics generally across the world, are used in animals, not in humans. So 70% of them, generally not for therapeutic use, are generally used as preventatives or for growth promoters. So you've got 70% used inappropriately in animals, and of the remaining 30% used in humans, about 70% of those are being prescribed inappropriately. So you've got this huge exposure of bacteria to antibiotics, which allows them to develop resistance other than directly when they're needed for therapy. So within Australia, in terms of antibiotic use in agriculture, we're actually one of the better countries in the world in terms of not unnecessarily using antibiotics in animals. However, with humans, we're at the other side. We're one of the top prescribers per humans in the world. So we do have work to do in trying to reduce unnecessary antibiotic use. So I pointed out earlier that every antibiotic resistance has developed over the course of time. But in the past, we've been able to keep up with the development of new antibiotics in order to come up with better treatments that can treat these drug-resistant bacteria. So it's been this constant arms race between bacteria developing resistance mechanisms and us being able to develop new antibiotics that can treat these resistant bacteria. So in the past, we were developing a lot of new antibiotics, and the problem is now that development has been greatly reduced. So we're no longer keeping ahead in the arms race with bacteria. And it's graphically illustrated in this, which shows the development of new classes of antibiotics, where for the past 30 years for gram-positive and gram-negative, and over 50 years for gram-negative, we actually have not developed a new class of antibiotics that's made it all the way into the clinic and being used for therapeutically. And this has led to what's been called a perfect storm. So you have this rise in number of drug-resistant bacteria accompanied by a sharp decline in the number of new antibiotics that have been approved every year. And this has leveled off now. There are about 3 or 4 being approved for the last 3 or 4 years. And the lack of new antibiotics is largely driven by the exit of almost all major pharmaceutical companies from antibiotic development. So from back here, you had about 20 companies, and there are about 3 or 4 that are still doing basic level research in developing new antibiotics. And the reasons for that are largely economic. So for a pharmaceutical company, it costs the same amount to develop an anti-cancer drug as it does to develop an antibiotic. You can sell an anti-cancer drug which prolongs your life sometimes only by a couple of years for $100,000 a year. And antibiotic therapy, which actually saves a life, it's very difficult to charge more than $1,000 for it. So economically, it makes no sense for pharma companies to invest in antibiotics. So we need to come up with new ways to try to reinvigorate the antibiotic pipeline and discover new antibiotics. So how can we do that? There are a number of different approaches. One of the most successful over the last 4 or 5 decades has been to rationally improve existing antibiotics. So that's why we have 5th or 6th generation beta-lactam antibiotics. You take an existing class, you try to make it more potent, you try to overcome the resistance mechanisms. Another approach is trying to rediscover old antibiotics. So during the 50s and 60s, it was the golden era of antibiotic discovery. So there are hundreds of discoveries of potential new antibiotics. Only some of those were developed further and taken into the clinic. So if you go back and look in the literature, potentially you can find these hidden gems which were reported at the time, but nothing further was done with them. There are efforts to repurpose old drugs. So there are some drugs for other indications which we're now finding can be used either in combination or on their own as potential antibiotics. Or you can try to use target-based approaches. And this is what industry was trying to do, particularly during the 80s, when you could identify molecular targets in bacteria which are distinct from human targets and come up with exquisitely potent inhibitors of those specific targets. The problem is then when you try treating an actual whole bacteria, bacteria have evolved all these mechanisms of resistance that prevent your compound from actually getting to the target in the first place. So target-based approaches in pharma have been spectacularly unsuccessful, and going forward, I think that's still going to be the case. And finally, you can try to uncover untested new chemical diversity. And this is where most antibiotics were originally found. So natural products diversity, people were testing extracts from fungi and bacteria and plants and discovered all the major classes of antibiotics. So there's been a return to this type of investigation using different, more modern approaches. But we also wanted to look at a different type of source. So there also is chemical diversity from synthetic chemists around the world, and I'll go into that a little bit more later. So we have examples of rational improvement, rediscovering old antibiotics, and chemical diversity. So I'll talk about an antibiotic development program that is looking to rationally improve an existing antibiotic. So going back to this graph of the rise of resistance, there are a couple of exceptions that have quite long periods before resistance developed. And one of these is vancomycin, which is a glycopeptide antibiotic for gram-positive infections. So as a starting point for developing an improved antibiotic, taking one where it took a long time for resistance to develop seems to be a logical starting point. So vancomycin interacts with lipid-2, which is a precursor for the synthesis of the peptidoglycan layer that forms part of the cell wall of bacteria. And lipid-2 is anchored to the membrane of the gram-positive bacteria. So our idea was to try attaching on some other substituents onto vancomycin, where you attach on this positively charged group, which can interact with the overall negative charge of a membrane, and also a lipophilic tail that can insert into the lipid bilayer of a membrane. So by doing that, hopefully you increase the affinity for the lipid-2 by providing this bidentate binding mechanism. So this is, I'll have a couple of chemical structures up here, but you don't have to pay too much attention. This is the vancomycin core. We attach on a linker, we attach on our positively charged, it's a peptide component, and then we cap it off with this hydrophobic lipid group. And so varying these 3 components, we did a structure-activity relationship campaign, made over 300 different analogs, and came up with some really promising lead compounds. So here are 3 of our lead compounds, a minimum inhibitory concentration table, and comparing their activity against several types of bacteria against commonly used antibiotics, including vancomycin, oxacillin, lamelazid, daptomycin, dalbavancin, teicoplanin, and telavancin. And you can see against MRSA species, we have activity as good or better than any of the other existing antibiotics. It's also active, retains activity against glycopeptide-resistant strains, so strains that are starting to develop resistance to vancomycin itself. And it also has really good potency against multi-drug-resistant strep pneumonia. And importantly, it retains activity in the presence of lung surfactant, where antibiotics such as daptomycin becomes inactivated. So we also looked at testing this against a larger panel of strains, so against 120 Australian strains. And the MIC90, which is the activity against 90% of the strains tested, again, was as good or more potent than any of the existing comparator antibiotics, including a relatively recently approved glycopeptide, dalbavancin. So one of the really critical things to evaluate when you're developing a new antibiotic is how quickly can resistance develop against it. So there are two types of measurements you do. One, you measure the propensity for resistance within an existing population of bacteria. So within 1 million or 2 million bacteria, 1 or 2 may already possess resistance to the drug. And doing that type of assay against both a Staph aureus strain and a strep pneumonia strain, our antibiotic showed very low propensity for pre-existing resistance. And the other type of study you can do is a resistance induction experiment, where you expose the bacteria to multiple generations of growth in the presence of a sublethal concentration of the antibiotic. So here's looking at over 20 days exposure to vancomycin, daptomycin, and cefepime, where you see this gradual increase in the MIC. And with our vancapticin compounds, you see a low but still very potent activity of the compounds. So from both those perspectives, in terms of developing it as a new antibiotic, it looks really promising. In terms of doing in vivo studies, so we do studies in mice, which are generally very predictive of what happens in humans. So if we do pharmacokinetic studies, vancomycin has a very short half-life, which is why it has to be dosed quite often or used as an intravenous drip. And comparatively, our antibiotics show a much prolonged half-life, which means they should be suitable for once daily dosing or once every couple days dosing. And more importantly, when you do a thigh infection study, an infection study in mice, so we inject bacteria into the thigh, let it grow for 24 hours, and look at the difference with treatment or not treatment of the antibiotic. So you can see without treatment, you get this huge increase in growth of the bacteria. Testing a set of antibiotics at the same concentration, so both vancomycin and linezolid showed no statistical reduction at 10 mg/kg dosing. Daptomycin, you see an effect, whereas vancomycin A and vancomycin B in particular, you see really strong activity. And if you take vancapticin B and do a dose-response study, you're still seeing an effect as low as 2.5 milligrams per kg. So it's very much more potent than vancomycin and works in vivo. We've shown efficacy in multiple models, so in an MRSA lung infection model, a strep pneumonia lung infection model, a bacterial sepsis model, and a peritonitis model. So in a variety of different indications, these compounds work very well. The other thing you have to be very conscious of in developing an antibiotic is its toxicity. So vancomycin, its dosing is limited by nephrotoxicity, damage to the kidney. So again, another in vivo study looking at kidney damage in mice treated for 7 days consecutively. Vancomycin shows typical signs of damage where you get this tubule dilation and necrosis in the tubules, whereas the treatment with our vancapticin B showed no damage after 7 days of treatment as well, at a dose much higher than needed for efficacious treatment. So the compound is scalable. This is what it looks like, a nice fluffy white powder. It's being produced commercially on a 10-gram scale by a contractor company. And the status of this now is under investigation for licensing by a small antibiotic-focused biotech company who are potentially interested in developing it further. So, the compounds are potent, they're effective in vivo, and they show reduced nephrotoxicity. So moving on to a different approach, the rediscovery of old antibiotics. This is focused on a class of antibiotics called the lipopeptides. So the polymyxins are currently being used as a last resort antibiotic. They were first discovered in the late 1940s, have a lot of nephrotoxicity effects, so weren't used very widely, but have, particularly in the 90s and currently, are reintroduced as last resort antibiotics to treat these highly multiple resistant gram-negative infections that no other antibiotic works anymore. The problem with colistin is that it's extensively used in inappropriate ways. So it's used in feed, particularly in China, in chicken farms. So you have these 40-kilogram drums of colistin being used to treat animals. So resistance is developing very rapidly, and there's been a lot of publicity, particularly over the spread of MCR1 resistance gene. So in the 1970s, there was a report of a structurally similar class of compounds called the octapeptins. And what attracted our attention to this was that they were reported to be active against polymyxin resistant strains, which was surprising because, again, their structure is very similar to that of the polymyxins. So we did the first synthesis of one component of these, reported last year, octapeptin C4, and found that its activity against a range of sensitive bacteria was not as potent as polymyxin B or colistin, but it retained the same activity or as more against these really highly resistant strains. And more importantly, in terms of induced resistance, so the same type of experiment I showed you with vancomycin, polymyxin and colistin, you get this huge increase, almost 1,000-fold increase in MIC over the course of 20 days, which is within the course of time of treatment within a patient, whereas octapeptin, you get a much reduced rate of induction of resistance. And importantly, there's no cross-resistance between those two induced resistance strains. So the strains that were induced to polymyxin and colistin retained sensitivity to octapeptin, and the strains that induced resistance to octapeptin retained sensitivity to the polymyxin. So they appear to be acting by a different mechanism of action. So we did the same thing, a structure-activity relationship study, identified a potential lead compound which has improved activity compared to octapeptin C4. It shows efficacy in a phi infection model, so now we're doing a gram-negative E. coli phi infection. And again, nephrotoxicity is a really big issue with the polymyxins, and we've done some human cell-based, so fresh cultures isolated from human kidneys, using a variety of markers that are predictive of nephrotoxicity. And you can see that gentamicin, polymyxin B, show really high signals for nephrotoxicity, whereas these new compounds appear to be, at least in vitro, much less nephrotoxic. So again, in terms of mode of action, we've developed some fluorescent probes, and they appear to be acting differently. So we have this potential new class of compounds, which we've just received a development grant for to take forward into further testing in order to try developing a potential clinical candidate. But certainly for something as a last resort antibiotic to treat highly drug-resistant gram-negative infections, we're very excited about this class of compounds as a possibility. And just to finish off quickly, in terms of trying to discover chemical synthetic diversity, other than natural products, chemists have made approximately 80 million compounds over the last 100 years. The majority of those have never been tested for antimicrobial activity, and they're sitting in shelves and labs across the world of academic chemists. So in one database, only about 14% of the million compounds in there have reported antimicrobial data. And in the past, antibiotics were originally discovered by collaborative efforts between people working together without having MTAs and CDAs and things that blocked the exchange of information and compound. So COAD, or Community for Open Antimicrobial Drug Discovery, is an attempt to return to a collaborative global effort. So it's a global initiative where we seek compounds from chemists around the world and offer free screening to test for antimicrobial activity. And we test against 5 different bacteria and 2 fungi. And we're able to do this because we have significant funding from the Wellcome Trust, so about $6 million over the last 3 years. So we test against 4 of the key gram-negative pathogens. We test against 1 gram-positive MRSA, and we test against 2 fungi because that's another growing area of concern in terms of there are very few antifungal drugs and resistance is developing. We have a website to try making it as easy to submit compounds as possible. It just pulls up a submission form, enter compound details, and we don't require any formal paperwork. All we need is like a software license. You click to agree our terms and conditions. We have a workflow where we receive compounds, dissolve them up, format them into 3 to 4 well plates, add the bacteria or fungi, incubate for 24 hours, and this measure growth or no growth by the presence of cloudiness from the growth of the bacteria. Reports generated, data gets sent back to the submitter. So this initiative has been highly successful in the last 2.5 years. 200 different academic groups from now more than 40 countries around the world have sent us over 200,000 compounds to test to see if they are potential antibiotics. In terms of the numbers received, so we're screened about over 180,000. Our hit rates, so the number of compounds that show activity, are significantly higher than if you were to screen commercial libraries. So it shows that the academic chemical diversity potentially does contain new antibiotics. And as highlighted in a Nature report last year, we've already screened twice the number of any other open access type of screening program in the world. So we're having real impact in terms of the people that are getting our data are using them for publications, for filing patents, and for grant applications. And perhaps more importantly, we're engaging researchers into antibiotic discovery. So potentially, we've done a survey that suggests there are less than 1,000 actually left doing antibiotic research in industry, and we're engaging more than 500 in the academic side of things. So we look as COAD as the missing piece of the puzzle. So you have these collections of compounds. You have these programs which take validated compounds and develop them into antibiotics. And we think that COAD fits in the middle to provide that additional data needed to bridge the academic chemists to these programs that develop antibiotics. So we have received global recognition. Dame Sally Davis, the Chief Medical Officer of England, is a strong supporter of us. And so we're hopeful that this early stage program will lead to the discovery of new classes of antibiotics. So I'll finish off there, and thank you for your attention.